Tonight, we have a kind of an exciting array of heavy hitters here. But before I introduce them, let me call on an educator, a young educator, who was the Dean of the College of Arts and Sciences at Syracuse University. She's a geologist. 23 years ago, she came to Syracuse as a leading paleontologist. And I know what paleo means. It means ancient, and I've been called a paleoconservative myself. But she's really built a College of Arts and Sciences at Syracuse that we're all very proud of. I was a dropout from Syracuse, and I'm very proud of that because I represent all the dropouts. And Dean Kathy Newton, you want to welcome the people?
Thank you so much, and it really is a delight to be able to co-host this series, Speaking of Science, in partnership with the Dana Foundation. One of the common goals between the Foundation and Syracuse University, and especially the College of Arts and Sciences at Syracuse, is that we feel we have very strong roles to play in incubating science that cuts across disciplines. And under our Chancellor's leadership and during my time as Dean, one of our foci institutionally at Syracuse has been cell signaling, of which we're going to hear many examples tonight. And so it's been a tremendous partnership with the Dana Foundation, which is making such tremendous strides internationally along so many different fronts. I am Bill Sapphire's dean, and that's a wonderful privilege. And I don't have an opportunity to do something like this very often. I do want you to ask you to join me in celebrating him. He's just received the Presidential Medal of Freedom, and he won't want us to do this, but may we have a round of applause for Bill? Thank you so much.
Okay, now let's get to work. This panel that we've got here, let me quickly introduce them because a short introduction, if they're big shots, always works. Bill Mobley, Professor and Dr. William Mobley is Professor of Neurology and Neurosciences at Stanford University, and he's Director of Stanford's Neuroscience Institute. His laboratory out in San Francisco studies the signaling biology of neurotrophic factors in the normal brain and in animal models of Alzheimer's disease and Down syndrome. He's been a consultant to Dana Foundation and helped steer us into some fascinating areas. And as a matter of fact, remember the movie The Graduate? When some old fogey took the graduate aside and whispered, "I'm going to tell you one word, plastics." Bill Mobley took me aside and at a recent gathering of scientists in San Francisco and whispered to me, "Circuits." All right? And that became the basis of this gathering. So he's here today, and I think he'll open our minds to the importance of circuits in the human brain. And we've brought from various universities several, as I say, heavy hitters in the world of circuitry. And remember that word because many of you here are scientists, and many of you are like me, total laymen. And this is the cutting edge of neuroscience. Mahlon Delong, he's Professor of Neurology and Director of the Comprehensive Neuroscience Center. Comprehensive neuroscience is neuroscience that comprehends everything at Emory University. His research has, I have to read this, clarified the functional organization of the basal ganglia, which, as we all know, is a group of nuclei in the brain. And he's described the role that they play in movement and in movement disorders such as Parkinson's disease. This man, who is a fairly modest, certainly very modest for a neuroscientist, he and his colleagues have helped clarify the diverse roles played by basal ganglia in motor, cognitive, and emotional functions. And his studies, he's a real pioneer here, have helped renew interest in the development of new surgical approaches to the treatment of movement disorders. The lady on the panel, Helen Mayberg, who is a professor of psychiatry and neurology at the Emory University School of Medicine. We've been following Helen Mayberg's career for a dozen years, and we're kind of proud of our association with this budding genius. Her long-term interest in neural network models of mood regulation in health and disease led to the recent development of a new intervention for treatment-resistant patients using deep brain stimulation. You'll hear the letters DBS here today, which is a study initiated at the University of Toronto and continuing at Emory. Brian Litt, member of the neurology and bioengineering faculties at the University of Pennsylvania. He's an active clinician investigator with a primary interest in the care and treatment of individuals with epilepsy, and studying how seizures are generated and how they spread through circuits in the human brain. He's an inventor and a medical entrepreneur, and he's co-founder of two companies, Intellimedics and Bioquantics. He's good at naming companies. And finally, the moderator this evening, and the one who conceptualized this gathering, is Guy McKhann, professor of neurology, neuroscience at Johns Hopkins University Medical School, and really one of the giants in the field of neuroscience. And we've been lucky to have him as a major consultant for us and in all our grants around the country. And he's also married to a professor who knows more about aging and the problems of aging than anybody that I know. And between the two of them, they're a great husband and wife team, and we were able to draw on their expertise in a variety of places. So that's the nicest thing I can say about you, Brian McKhann.
It's all downhill from here.
All right, now over to you.
Thank you, Bill. Well, good evening. I'd like to put what we're going to talk about in some kind of perspective here. About 150 years ago, a French surgeon, actually, was asked to see a patient, and the patient's name was Tan. And the reason he was called Tan was that was the only word he could say. He seemed to understand what people said to him, but no matter what they said, his answer was Tan. And Broca was very interested in where language was and how it was organized in the brain. And Tan died, and Broca found that he had a lesion right here on the left side. And he very promptly reported this to one of the French groups. They don't fool around over there. He did an autopsy, and 2 weeks later he gave his report. He gave his report in French, and I'm not quite sure the title and how I translate it. I think it was something like On Language or something like that.
Great thinking.
Broca made the observation, we speak with the left side of our brain. And that clarified a lot of the thinking at the time. It also started the era of localization in brain. Now, if we skip forward 100 years, that's when I first came into neurology, plus or minus a few years, and we were still stuck with that same issue. Where are things localized in the brain? And we spent a lot of time fussing around trying to figure that all out with whatever tools we had. In the last 20 years, our thinking has changed. It's changed to this idea of circuits. That there are circuits that are involved in processes, circuits involved in how we process vision, circuits involved, as Mahlon will tell you, how we make a motor movement. As Helen will tell you, circuits involved in mood. This is where things are going, and that's what we wanted to essentially focus on tonight. Bill Mobley is going to start by talking a little bit about how we study circuits and why it's important, and we'll go from there. Let me just set some ground rules. I've asked everybody who are speaking up here, we don't want a series of lectures. We want a series of conversations. So we would like people to have their formal remarks relatively brief, 10 or 15 minutes, so there's time for discussion after each one. And please, no question is too dumb. We get our best ideas from dumb questions. We really do. So with that background, Bill, I'll ask you a dumb question.
Well, thanks. This is great. Thanks to Bill, and thanks to Catherine, and thanks to Guy. Are there any students in the room? Just if you could raise your hand. So here's the deal. One of you is going to have to ask the first question. We're going to let the student, and another one is going to ask the second question. So get your questions ready, because here we go. Now, do I just push this? Well, this is a fun thing to think about. I am immensely excited about this organ we call the brain. It is, in fact, a defining organ. It defines us. What we think and feel, our sense of who we are, how we respond to our world, all of these are manifestations of our brain. And it follows that disorders that compromise the brain redefine us. So we're now at the beginning of an era. So the talk is basically the following. We're at the beginning of an era where it's possible for the first time to understand the brain, and by so doing, to understand ourselves. This is immensely exciting. The technology is in hand, or nearly there, that will allow us to understand for the first time how the brain actually works. Very, very exciting for all of us. Let me go back. Get to do this. Yeah. This is the ultimate frontier for human discovery. And I would say that as a result of this discovery, we are going to have the possibility not just of understanding ourselves, but of changing the world in which we live. What we learn by studying the brain will change fundamentally every aspect of our existence. We will change who we are as a society as a result of brain science. Now, cardinal principle is that neuronal circuits mediate all brain functions. Everything that happens, what you hear, what you see, how you behave, how you reach for something, your impression of someone, whether you feel good or poor, whether you feel, ebullient or sad, all those functions are in fact wired in circuits, very complex circuits to be sure, that all brain function is mediated by circuits. All circuits consist of neurons that are linked to one another. Here's a 3-neuron circuit. Here's the cell body of the first neuron, its nucleus. Here's a little antenna called the dendrite with a little spine right here. I'll show you some spine soon. And here's this wire called the axon that links neuron 1 to neuron number 2 at a point called the synapse. Information comes into the brain, is processed, and a response is made. Receiving, processing, and sending information is what the brain does best, and it does it better than any machine or any other tissue in the universe. It's a fantastic information processing machine. Now, this is the synapse, just very briefly. Here's that axon sitting here with some little vesicles sitting here waiting to be discharged. Here's a space called the synaptic cleft, and here's that next dendrite, that next antenna waiting to be excited by this chemical that flows across and activates its receptors. Circuits and synapses. All brain disease, every single disease of the brain, every disorder is due to circuit disruption. neuronal circuits are disrupted in disorders of the nervous system, and it might be at the synapse, it might be at the cell body level, it might be in the dendrite. In the end, all brain disease is circuit dysfunction. So how are we going to do this? How do we explore neural circuits to understand brain function? I think we have to think about the dimensions of the problem. Here's our brain. I've portrayed here that cognition sort of sits up here, and in the northwest and in the southeast, we start with molecules and cells. In fact, discovering and deciphering the brain is a puzzle that has to be pursued across dimensions of time and space. And the dimensions are huge. Times ranging from decades to milliseconds, 10 to the 9th milliseconds in a life, in a decade. And across spatial distances, they're also 10 to the 9-fold, from nanometers to meters. This is exciting, but it's profoundly difficult. This will take immense effort to go, if you will, from molecule to mind. And yet we can do it now. And we can do it now because of an amazing collaboration where we link the top-down guys, the cognitive systems neuroscience guys, with the bottom-up people, people like me who study molecules and cells, and mixing in, as Catherine was alluding to, these wonderful engineers, these wonderful physicists, and these wonderful colleagues, both clinical and non-clinical folks, that care very much about exploring neural circuits. So what's going on now? We're using existing tools and developing new ones to identify and analyze circuits. We're creating a theory that we need to link different levels of analysis. From molecules to cells is a big jump. From cells to circuits is a big jump. From circuits to cognition is a big jump. And finally, we'll use these tools and concepts to understand and treat brain disorders. So let's envision it this way. We have the synapse here, we have the brain here, and we have levels of analysis linking each using theoretical approaches and real data. We have a scale of machines that is also daunting. Here's a 10-ton imaging device. It's 5 meters across, huge. Here, a confocal microscope, and here a little platform that is 0.0005 meters across and weighs far less than a gram. We need machines of various dimensions to study circuits effectively. Now let's look at just a few examples at the molecular level. Very important. This guy talks to this guy. The information doesn't just flow from the axon to the postsynaptic area. It flows from the postsynapse to the axon, and the information that's sent is information that keeps circuits intact, that keeps synapses healthy. And one of the kinds of information that's sent are called trophic factors. These are trophic signaling proteins. They're made postsynaptically, released, bind to their receptors, and that whole complex is engulfed and transmitted down the axon to the cell body. This is signaling locally. In fact, all signaling is local, just like all politics. Somebody said that once, Bill. Local, spatially controlled signaling, very important. I now can watch this happen. I can watch this process happen in a living axon using a special compartment. I can modify that trophic molecule and put a label on it, stick it into a chamber, and watch it move down axons. In the next slide, I hope will show you that. In this slide, the ends of axons are here, and there are several cell bodies here, so it's going from the tip of the axon back to the cell body. And watch those little dots, if you will. Right, watch the dots. Every dot is an NGF molecule. Every single dot is contained in a tiny vesicle called an endosome. And those dots are moving in regular order through the darkness, through the axon, to cell bodies located over here. This is a real-time movie. We can now watch a single endosome move with the precision of a tenth of a micron per second. A tenth of a micron per second over hours and hours. Very exciting. Now my movie won't stop. You know, when I want it to stop, when I want to start it, it'll never start. Here we go. Let's try this. Let's work up one level. Let's look here at information, new techniques and technology going on at the levels of synapses and interacting neurons and circuits. And let me turn to my colleague Stephen Smith at Stanford. What he does is he takes a piece of tissue, cuts a very thin piece of it, and lays them out in ribbons, and then is able to stain these with antibodies. He can then reconstruct these thin sections together, and this is a piece of cortex. And what you see in the green are the axons that are coursing through the tissue, and what you see in red are individual synapses that are being made upon cells near those axons. Incredible beauty. And Steve can now do this, not just with a mouse brain. He can do it with an entire human brain. He can reconstruct in 3 dimensions an entire human brain, and happily can stain with as many as 50 or 60 different antibodies. He can populate a synapse with 50 different markers. An amazing advance. Here, at a slightly lower magnification, one sees a cell body in the cortex. Look at this beautiful green cell. This is a neuron with its nice long dendrite here. And in the neuropil, in the space around it, are these little red synapses making hundreds of thousands of contacts, even in that one tissue slice. And here at a higher power, here's one dendrite, one of those antennae. Here are those spines I told you about. And notice the beautiful co-localization of these synaptic molecules with those spines. Amazing clarity and amazing possibilities. What about the structure of circuits in the brain? This is work that Mark Schnitzer's doing at Stanford. One of the problems we have is how can we look deep in the brain? With 2-photon microscopy, you can't really do that. Even with confocal microscopy that goes deeper into the brain, you have problems. We want to see the hippocampus. We want to see the basal ganglia that Mahlon, we want to see the striatum, places where Mahlon works and where Helen worked. How are we going to do that? So let's actually invent a new microscope. This is called fluorescence microendoscopy. This is a fluorescence microendoscope. Yes, that's a dime. It's as big as a dime, fits on a mouse head, and the mouse can wear this for months at a time. Light comes in red, and light returns in green, and now you have a 2-photon microscope that sits deep in brain tissue and takes beautiful images like this in hippocampus. Here are those cell bodies in hippocampus lighting up with their beautiful dendrites. Can you make circuits work differently? Can you turn circuits on? You can do so by stimulating circuits, but you can also do so using light. And here's work from Carl Dijsselroth. He took a little channel from a little algae. And this channel is very important for helping the alga figure out where it needs to be with respect to the sun. It lets a little current through this channel when the sun shines on it. Carl took that channel and put it in the brain. And now neurons that are infected with that channel respond by firing when you shine blue light on them. So this is an optogenetic method for circuit activation. Carl now has the ability, using special DNA constructs, to put his little channel in any population of neurons in the brain he cares about. He's finding modified channels, so pretty soon he'll have a whole set of flavors of channels. He could literally sit and shine light in the mouse brain using Mark's microendoscope and play, like on a piano, play the circuits of the mouse brain. It's coming to a university near you very soon, I promise. How about up here in the level of cognition and behavior? My last couple slides. You've heard about MRI, functional MRI. Here's the beautiful picture that one can make of the back of the brain when shining light on a certain part of the retina. Here's the middle of the retina, and here it goes out more eccentrically. Brian Wendell's beautiful work, making visual maps. There's another really terrific tool, and you hear about it. We can now measure and map the tracks of circuits in the brain in humans without any invasiveness at all. People sitting in a scanner can now be imaged in such a way that we can find the axonal tracks. And the reason we can is that water moves far better and faster down axonal cylinders than across axons or from one axon to another. And so one can use this data to construct maps like this. Here's a human brain dissected, showing all these axon tracks that are covered with white matter, with myelin. Here, exactly the same technique as I've just mentioned, this tractography, can now be used to image different parts of axonal tracks going to different parts of the brain. So quite soon, it should be possible to be able to physically show the connectedness of two different brain regions, or even more than 2 different brain regions. So a lot of progress here, a lot going on, and a lot to happen. Those are my comments. I think that imaging neural circuits, working across scales, that's where we are. There's immense possibilities for progress there, and quite frankly, you ain't seen nothing yet. This is exciting. Thank you.
Thank you. sit over here, Bill. Where's our student? Yes.
Yes, that was a really intriguing talk. I was wondering what your thoughts are on where you think, like, what do you envision being the cures for instance, Parkinson's and depression and epilepsy?
Right, yeah. So let me just say, I wish I could just give you, one sentence answer to that. What I'll say is this. I think the cure will be found in the circuitry. I think we'll understand the circuits for the first time. That knowledge will guide us to understand circuits that are abnormal in diseases, and we'll have the tools we need then to fix those circuits. Ultimately, we're going to have to repair circuits to restore function. Great question, and we're not there yet.
Bill, what's the limiting factor? What's going to keep us from getting there?
I think one of the limiting factors, a terribly important limiting factor, is the ability of the need for a culture that encourages crosstalk between disciplines. Just as you were speaking about before, our stuff on moving individual NGF molecules was impossible without working with Steve Chu, who's a Nobel Prize-winning physicist at Stanford. Steve and I are really good friends, and we didn't start out really knowing each other, but we sort of got interested in one common theme, started to collaborate, and it's changed all our work. I needed Steve, even though I didn't know that. By the way, I used to think that scientists had really interesting questions that they couldn't answer, and they went to engineers, and engineers invented machines. I know that's wrong. Technology happens. Technology happens. It hits you sometimes in the head, and you don't even realize that now this tool that you always needed, well, it's here, and now it's your chance to use it. So I would argue one of the biggest limiting factors is finding cultures, building cultures that are generous with respect to funding, that encourage risk-taking behavior, that stress technological advances, and that build bridges between different disciplines. That would be a huge step forward if we could do that. And I mean by that, engineers, physicists, poets. I'm going for poetry here. Poets are very important people. I don't have a lot of friends that are poets, but I can just tell you that I love poetry. It's good for my brain. I need some more poets in my lab.
Well, all right, I think we'll move on. Bill has to leave us because he's catching a plane to Germany in about an hour or so.
Yep.
Less. So, Mahlon, while they're hooking you up, Mahlon is the father of a whole field, which essentially started with figuring out the circuits, the kind of circuits that Mahlon was talking about, but how they applied to control of the motor system. And once he figured those out, he went and he looked at how he could use them in an animal model of Parkinson's disease. And from there, he went to devise therapeutic surgical approaches, which are now used all over the world. One of the things that we all have in common, that I should mention perhaps, all these people made two very wise decisions in their life. The first one was to take part of their training in the neurology department at Johns Hopkins. And the second was to leave. But Mahlon did his first work when he was in our department. And he came in one day and he said, Guy, we've been looking at the wrong target. The target we should be looking at is the subthalamic nucleus, a very small group of cells. And I said, Mahlon, that's crazy. Get a grip. No way in the world is anyone ever going to hit that nucleus in a human being. Well, we figured out that there were something like 14,000 operations done in the last few years, so I think I was probably wrong. Mahlon, tell us what you're doing.
Thank you, Guy, and certainly delight to be here and share the stage with people doing such interesting things. I'm going to try to turn my computer on here before. There we go. This topic is one that I find very fascinating. Let me make sure I can, at the same time. Oh, this was a glitch. Though. Yeah, okay. I'm going to talk about circuits and circuit disorders. I think it's obvious what a circuit disorder is. It's a circuit that doesn't work. and it produces some kind of disturbance, and it may be a disorder that we understand and define and recognize, like Parkinson's disease or obsessive compulsive disorder. So that's what we mean by a circuit disorder. These are the structures that we're talking about. I'm talking about primarily, and these are the basal ganglia. These, I think, are probably some of the most interesting parts of the brain because they seem to be not only important for behavior that's, we understand as movement, but also for cognition and complex activities of behavioral type. They're called basal ganglia because they're ganglia, the old term for nuclei, and they're at the base of the brain. So you can see here I've done myself in again.
Black button.
Yeah, there we go. Okay. These are small structures. Some of them are small structures. Got to do this right now. The subthalamic nucleus, the guy mentioned, is now former bit player, now center stage. The globus pallidus, the external internal segment, the putamen, and of course the caudate. These structures seem kind of randomly organized, and the thing that links them really is their connectivity. So let me jump to that. And the old ideas about it were certainly greatly poorly. The ideas about it were poorly formed because there wasn't really good anatomy, good physiology. 1970, Kemp and Powell formulated what I think became kind of the final word. It seemed like the final word. It was that the basal ganglia were there to take all the information from the cortex, from all those diffuse, diverse cortical areas, integrate it and send it to the motor cortex. It was believed that movement was driven by different cortical areas. The frontal lobe could initiate a movement. The sensory areas could initiate a movement. The visual areas. It was kind of an interesting idea that the cortical areas didn't talk to each other very easily, and they had to use the basal ganglia. That always bothered me. And when we began doing work in the basal ganglia and looking for areas that were related to movement, we found that they were not at all widespread. They were really very focused in specific areas of the basal ganglia, particularly in the pallidum. That suggested there was a whole lot of leftover pallidum that might be doing other things. And it was very easy to figure out then that, just reviewing a lot of the anatomy that existed, that we needed a whole different scheme. And that scheme was that of parallel segregated pathways. This depicts now schematically here the diversity of inputs to the basal ganglia, here the striatum, the caudate, the putamen, and then the output nuclei, the pallidum and nigra. The inputs come in from diverse areas, motor areas, eye movement, areas for eye movement, frontal areas for behavior, so-called association or areas, and areas involved in emotion and motivated behavior, so-called limbic areas. And the important point from this scheme, as it differed from previous, was that the pathways remain relatively segregated from another. So the limbic eye movement motor areas were traversing different parts of the striatum, different parts of the pallidum and the nigra, and different parts of the thalamus, and returning largely in a closed-loop manner to their sites of origin. And here you see schematically some of these frontal areas that were identified in the first work. This was work with Peter Strick, Gary Alexander, when we were at Hopkins, or at least two of us. You can just see the colored areas there. So now we've broken apart, we've sort of taken out basal ganglia as being components of larger networks, larger circuits that are engaged in various kinds of behavior. Now, I think it's reasonable to talk about circuit disorders as being basal ganglia disorders because they all engage the basal ganglia, but one could think about it just as circuit disorders. But here I'm using the term basal ganglia circuit disorders because there are other disorders that do not engage the basal ganglia. But certainly Parkinson's, dystonia, Billismus, chorea, and even Huntington's All of these, I think, represent clear examples of movement disorders that result from disturbances within the motor loop, if you will, or motor circuit of the basal ganglia. We now understand from imaging and other studies that neuropsychiatric disorders, Tourette's, obsessive compulsive disorder, depression, as you'll hear, all of these may really be understood as also disturbances of neural networks outside of the motor loop, at least in part, and share a common substrate in that sense. This is a way of looking at these structures beyond what we really want to focus on, but here you see cortex to striatum, processing internally within the basal ganglia, all those shaded areas. We talked about an indirect and a direct pathway to the output nuclei, which then return via the thalamus to the cortex. There's also output downstream, but for most part, this is a circuit. So all kinds of areas where there could be problems develop in both this direct and indirect pathway. And it turned out that one of those played a particular role, as you've already heard. Well, if we talk about hypokinetic disorders now, we think of Parkinson's disease as a prime example, and I'm not going to try to dissect that too much. The cardinal features of this are well known: slowness, the akinesia, tremor, difficulty initiating movement, and muscular rigidity, but a host of other disturbances that represent deficits resulting from more than dopamine deficiency. Dopamine deficiency largely accounts for these areas over here. We were able to understand some of the changes in these networks by recording individual neural activity in the basal ganglia in patients with, or I'm sorry, in animals, these are primates with Parkinsonism produced by neurotoxin MPTP. The interesting finding was that the activity changed dramatically throughout the basal ganglia in ways that were quite consistent with increased firing in the output nuclei and altered firing, as we see here, to patterned oscillatory activity. And also in the subthalamic nucleus, there was a dramatic increase in firing and change in the pattern. This was not obviously going to be the case when we started, and it surprised a lot of us that it was so obvious, these changes, and so consistent. What came out of this work was a strong reinforcement for the idea of a rate model of Parkinson's with excessive outflow from the basal ganglia, which was inhibitory. And this inhibitory outflow suppressed activity in the thalamus and cortex and even perhaps the brainstem. And the culprit, the key to understanding all this, turned out to be this somewhat overlooked subthalamic nucleus, which seemed to be firing and driving the output, because this was an excitatory glutamatergic output. So we thought we understood some of the circuitry and could explain a lot of the findings. The rate model has really fallen a little bit out of favor because of recent findings and some of it anticipated in the early work you saw there, that actually ensembles of neurons seem to be discharging in frequencies that are quite abnormal. In normal basal ganglia and motor areas, there is a high frequency activity in the gamma spectrum over 60 hertz, whereas in Parkinson's, these are replaced by lower frequencies in the beta range. And so there seems to be something happening with these local field potentials that we record throughout the entire network. So this is a slide now that will try to illustrate in many ways the problem and the treatment. Here you see the same familiar structures, but now arrows pointing to problems and to opportunities for treatment. In Parkinson's, we have a loss of dopamine, everybody knows, so we can replace dopamine and give precursors, and we can use drugs that modulate dopamine. Those act in the striatum. We can give trophic factors that may play a role into the replacing some of the neurons and supporting their activity. We can also lesion the subthalamic nucleus or stimulate it. And that was one of the key findings that Guy was mentioning that we had done in our models and found that that was extremely effective in reversing Parkinson's. We can give glutamate antagonists to block that overactivity all along the pathway here. We did pallidotomies for many years after we found this out, because that was also very effective, but had limitations. And then thalamotomy as well. And then deep brain stimulation, when that came on the scene, was then could be directed to these same targets. Turned out to be very effective, and a way to modulate activity without doing actual physical damage or minimal physical damage to the structures, a reversible approach. I come to functional neurosurgery because I think it's evident that that is the exciting area that, of our time. Classically, this was driven by a lot of luck, serendipity, and sort of somewhat hunches. But more and more this is being guided by our understanding of networks and organization from all of the approaches you've heard about. Animal models have played a great role, particularly the MPTP primate model, which has helped us to really identify new targets. I mentioned pallidotomy, which means simply interrupting that circuit at a key point, and that is in the motor part of the circuit. That is a very effective operation. All of the features improve in the motor features, contralateral to the side, and lesion location size are major factors. And the lesions must be placed squarely in the sensory motor part of the pallidum. I can show you lots of data to support that. But there are disadvantages. It's a lesion that is large. It's an irreversible change in the network. And there can be side effects and damage to structures adjacent with just millimeters away that are not desirable. So the arrival of deep brain stimulation was greeted with great enthusiasm as a way of modulating in a reversible manner these same structures and networks. Being able to adjust stimulation frequency, voltage, and duration. And certainly, on the other hand, just to turn it off and go back to square one. Oops. I think that. How does DBS work? There are many theories and evidence to support considerable range of activity or effects. It could inhibit cell bodies, could stimulate efferent, afferent pathways, could deplete neurotransmitter, could replace abnormal patterns of activity with more normal or more tolerable patterns of activity. And also to think about being able to just release the otherwise normal areas, intact areas of brainstem and cortex and thalamus from this abnormal influence. Probably all of these play a role, but maybe the most important is this replacement of the abnormal patterns of activity throughout the network with a more tolerable pattern. DBS is done very much like you're going to do an ablation, except that you, rather than burning or coagulating, you insert an electrode with a number of contacts and connect that up to the pulse generator implanted in the chest. All of this is very much like a cardiac pacemaker, simply implanted subcutaneously. And there you see it, and you can stimulate through any of these. Here we see schematically how it might be placed in the subthalamic nucleus and how it's possible to stimulate either between two of these contacts or any one of them and modulate the activity in that structure. Here you see an MRI with the actual image. This looks horrible because of the artifact, and it's really much smaller. It's about 1 millimeter in diameter. And here you see on the other side a pallidotomy. So you can combine these procedures. I'm going to just say a word about dystonia because I think it's probably one of the most interesting of movement disorders. And I want to, rather than show you a patient with Parkinson's who's had DBS, which you've all probably seen, I want to show you one with dystonia, who I think will give you another perspective. These are patients have involuntary movements affecting the limb, any body part, and often with a lot of co-contraction and overflow with attempted movement. You have to see it to really understand it. Words are inadequate, and you'll see one in just a second. Like Parkinson's, this is a circuit disorder, and we've recognized, can now characterize the activity as being similar to Parkinson's in some way, but the rates are lower. But in other ways, it has a lot of the features in terms of oscillatory activity and abnormal patterning. We think there's overactivity in both the indirect and the direct pathways, leading to a decrease in activity with this abnormal pattern. But the targets for this, for intervening, are very similar, if not identical. As I said, this is an example, I think, of a circuit disorder. The interesting thing about dystonia is that the effects of stimulation or ablation are delayed. Days, weeks, months to see the full blossoming of this effect. So we believe this really relates to a fundamental change in neuroplasticity within the basal ganglia. And our effects are, in a sense, mysterious because we don't really understand what this really is all about. But there's a great deal of evidence for this fundamental disturbance as part of this disorder. It would be impossible to acknowledge all of those who have contributed to this work, and I have told you really the tip of the iceberg, but let me just single out Tom Wickman, some of the basic work, more recent years, Jerry Vitek, and a lot of the functional surgery and some of the basic animal work, and then a number of other colleagues here who have mentioned Hagai Bergman in particular, and others, all listed there, who have contributed greatly to this. I want to show you now a video. Let's see if I can.
I love cats, I love animals, and I can finally take care of them. The cats are new. Cindy and I both love cats a lot, don't we?
Oh, yeah. One thing, people wouldn't think, well, it's not that hard to hold an animal, but before the surgery, it would have been.
My tremors and my muscle spasms would have been overwhelming for me. It would have been dangerous for the cat.
Like you're going to drink first.
Peter Cohen's dystonia, which began with a tremor in his hand when he was 18, had begun to overwhelm him by the time he was married. He'd had to give up practicing law.
I was basically incapacitated. I couldn't sit, I couldn't walk, I couldn't stand, I couldn't even sleep really. I had constant tremors. I would eat on the floor. I had basically, no life for a long time. When I walked hunched over and my head was tilted down to the left like that. And people would obviously gawk and stare at me. People would make fun of me. That was a very large emotional drain for me to have to deal with that every day.
Peter was in a lot of discomfort, and actually when first time I saw him, as I came in the room, he was lying on the table, and obviously in somewhat contorted position. It became evident to me within a few minutes that he was really very much needed the surgery, and it was not a matter of if, but when. And I think I was pretty forceful in telling him he had to take that step.
I was a little bit nervous, but basically I had no other options. It was my last real hope to get better.
How does that feel?
It feels okay. It's been a long process, but gradually I would find that I was able to do things I couldn't do before. I could sit, I could drive without holding my head up, for example. I could walk great distances. So gradually over time I had more and more things happen that were great for me. A lot of people say to me that they didn't realize how tall I am. I'm oftentimes almost bumping my head on various ceiling type things because I don't realize now how tall I am. It's great. But I couldn't hold my head up before. I couldn't hold on to the handlebars with my right arm. Now I can. So it's a great feeling of independence to go outside and go for long bike rides. I love nature. I love being able to see nature. I couldn't do it before.
Yes, ma'am.
You were talking before about bridging the gaps across disciplines, but my question is, what about bridging the gaps within medical field as well. How these relate with epilepsy? How is related to? Is the same problem in different parts of the brains, or is completely unrelated, or we don't know?
Can you? I didn't understand.
So the question was, how is it related to epilepsy across the same discipline? It might be fair to say I'll talk about that at
yeah, he'll talk about that.
But it was a great question.
Good.
Great question.
Mahlon, how commonly is this done now for Parkinson's disease?
For Parkinson's, this is kind of standard fare now. Patients who have lost their benefit from medication, that's not quite fair. Patients who experience a lot of the side effects from medication, or whose tremor does not respond to medication, for example, or who can't tolerate the drugs because of the cognitive and emotional side effects, those are excellent candidates for surgery. The benefit from surgery is exactly what the best that can be seen with medication. So you can tell patients, or show them, you're so much better when you're taking the medication, but then you have dyskinesias and the other problems. The surgery will give you the best without all the side effects. It's 24/7. You know, you don't have to take the pills. You don't have to, well, you do have to take pills, but you are now getting a more even benefit from the treatment. All right, guy.
How long it lasts?
We have seen no loss of benefit. That's the interesting thing. Not to say Parkinson's is not a progressive disorder, and there are aspects that are not tied to dopamine deficiency, but in terms of the dopamine symptoms that are dopamine deficient, I think we're quite impressed how long-lasting this is.
Isn't it true that there are patients that can go off medication?
Some patients with bilateral stimulation, and I could show you a picture of one who is running up a hill off medication, but generally speaking, at some point, they need to be on a little bit of medication. And I tell patients that's not the goal, because then they'll be disappointed if they're not completely off medication. I think it's to get function. Right.
Give me for being a policy person, but is it covered?
Oh, yes. Yeah, completely covered, yeah. Well, that's a very important question, yeah. This has been covered for many years now by Medicare and also private insurers. Yeah. Dystonia only recently, has become covered.
Dystonia sounds like a nation in the Baltics. It's a terrible thing that is not getting much publicity.
I'm glad you said that, because I work with a foundation, the Dystonia Medical Research Foundation, whose biggest challenge is really overcoming this lack of awareness. This is the third most common movement disorder. I think it's not appreciated because of not its name, but the fact that it can appear in so many different ways, that patients who have this affecting their eyes are seen as very different from those affecting their speech or their jaw, and those who have writer's cramp or musician's cramp. It can appear focally on almost any part of the body, or it can be generalized in children, but it is all added together is a huge problem, and one that very much deserves more study.
Okay, I think we need to get on. Okay, Dr. Mayberg has been on a very interesting interface. It's the interface between neurology and psychiatry. You would think that those two fields would be very intertwined and very interactive, but historically they haven't been. And part of the reason has been because for a lot of the diseases we think of as being psychiatric illnesses, we don't have much of an idea of what's going on in the brain. They don't have a pathology. Parkinson's disease has a pathology, and that led to how to get at it. But if you look at the brain of someone with depression and they die, that brain looks pretty normal. If you look at the brain of someone with schizophrenia when they die, that brain looks pretty normal. So it's been very hard to get a handle on what's going on here. Helen has gone at this more as a neurologist would, trying to get at the issue of, okay, what's the matter with the circuits? And can we do something about it? So she kind of is taking the approach that Mahlon took to a different area. Helen, you're on.
Thank you. I'm really delighted to be here, and particularly to be with my colleagues and my teachers. Not to date anybody, because I think with time we all kind of converge, and we all seem to be the same age. It's non-linear. I'd like to just start by acknowledging my collaborators, and mostly my collaborators in Toronto, who really allowed me to try out this idea that we've been thinking about in a very kind of diffuse and non-formed way for many years by our neuroimaging work, and also funding, and more recent funding now from the Dana Foundation to continue these studies at Emory. I think what's important, because we're moving from a very comfortable zone of neuroscience of we know exactly where we are. We have these cells, these synapses, these chemicals going down little channels. It's all very concrete and very believable that the brain is the brain. And when we move into a psychiatric disorder, it's suddenly much more fuzzy. Psychiatry thinks it's moving into biology when it talks about chemical imbalances. That's a Herculean and almost seismic step away from psychodynamic views or something with your mother that caused the illness. And it turns out it probably is something to do with your mother in terms of genes and early life experience. But in fact, the concept of psychological to chemical, that you're sort of a quart low on transmitter X, really is inadequate. And I just, instead of coming at it as Mahlon did by let's study motor systems, define it, and then bridge it to a disease and look for points of intersection where pathology is known, psychiatric disorder, you have to start much more loosely. And let me tell you, that's very hard for a neurologist. But I want to pose the notion in terms of the clinical need for a new paradigm in this disorder by just the nature of the problem. So it's now considered by the World Health Organization the leading cause of years lived with disability. This is a disorder that starts in young adulthood. So even though other disorders are very devastating, this one, unless you take your own life, doesn't kill you. And in fact, you wallow for many years in terms of your years of most productivity. The incidence is now estimated at 7%, and if you translate that into the United States, that's about 20 million people at any given time in a year will be afflicted with a major depressive disorder. And the estimates that are about 10% of people are actually truly treatment resistant. That doesn't mean I didn't want to take a pill, or I didn't take a pill long enough, or I didn't like my therapist. That's a systematic treatment of multiple classes of medications or psychotherapy. And that is really a huge number of people when you look at the incidence of the disorder. And critically is we do have a well-validated evidence-based treatment, electroconvulsive therapy. It's extremely effective in people who have quote-unquote drug resistance, therapy resistance illness. But the data actually says that once you've been through about 4 different medications, your chances of actually getting better with ECT and staying well is extremely small. So in fact, that is a very sobering statistic. And there are certainly a number of new developments. RTMS is being evaluated, I think, by the FDA in 2 days. Vagus nerve stimulation was approved last year. But the response rates are actually low in people that are in this ECT resistant category, which means if you don't take your own life and the suicide rate is extremely high, you have 0 options. Now, as Guy had said, the problem is we don't have the spot, we don't have the pathology, we don't have the chemical, we don't have the money and the bullet as to where to go. So how we develop new treatment strategies tends to be serendipitous, or tends to be based on Me Too strategies. Another Prozac, a Prozac plus another chemical, a tricyclic antidepressant that isn't as toxic or with as many side effects. And so conceptually, the court low on your favorite neurotransmitter needs a new approach. Now, we've worked for a number of years, and this work started actually at Hopkins, working with a psychiatrist who was interested in stroke depression. The model led by Broca, there's an interesting observation of people who have strokes, depending on where the stroke is. Not that you're depressed, because wouldn't you be if you couldn't talk and you couldn't move your arm, but that actually particular lesions in the brain have a higher association of people developing a major depressive episode than others. And Bob Robinson really articulated that there was something special about your frontal lobe and your basal ganglia, particularly on the left side. And I got involved in his group. And there was also evidence in Parkinson's disease and Huntington's disease, in many of these disorders of the basal ganglia, that they had a disproportionate high association of major depression. Actually, not as a result of the illness, but in fact, many of the patients actually presented with the depression, and then the rest of the movement disorder showed itself. And this was extremely interesting from my point of view, because it was really evidence straight from neurology of the idea of there were lesions or systems in the brain associated with production of mood symptoms. And instead of it being this sort of nebulous set of signs and symptoms, it could really be approached like Parkinson's. We, over many years, went to study that. And again, I think Bill Mobley said it extremely well. You exploit the availability of tools that you have. And I grew up in an era when I trained as a medical student, we didn't even have a CT scanner. It came in when I was a fourth-year medical student. And so you live in a time where you watch and have availability of methods that allow you to test these principles out. And if we hadn't had those, nothing that I'll be describing would have been possible. It would have really been reckless, because all we would have to go on is experience of psychosurgery, making lesions as the impetus of what we might want to do, which is a standard that, at least to me, was not acceptable. But imaging allowed us to actually articulate the nodes in the circuit in a much grosser and much less elegant way than Mahlon described, but in patients through functional imaging. And what we did as mostly a bookkeeping for a while, what areas of the brain are abnormal in patients with depression, studied with PET scanning, looking at resting patterns of activity and really looking for the functional lesion, if you will. And by doing studies of treating people with very conventional treatments, drug, therapy, placebo, in a number of different experiments, found that there were a set of brain regions that we could organize exactly like circuits that Mahlon described, except that they were really an architecture of mood regulation. And that, coincident with many other kinds of experiments, the brain regions involved in depression are really a constellation of a disturbance in mood and a disturbance in thinking. And it really is the interaction of emotion and cognition, with also disruption of circadian systems. And we know about where those kinds of behaviors live loosely. Again, I can say it now because Bill Mobley's gone. You get down to that microscopic, watching those little things move down the axons. I mean, I'm in awe. I want to be a neuroscientist when I grow up. But in fact, that we can articulate and actually build a circuit of depression de novo from the behavior through imaging and test some principles. Now, one of the things that we started to notice over the years was that certain areas of the brain seemed to be the more major players. And for the sake of time, what we found was that as you treated people with various treatments, our studies of just classic antidepressants, your brain with depression, your brain on Prozac, what's it look like when you get better versus when you don't, your brain thinking it's getting Prozac, getting placebo, getting better. Well, we start to notice that there were two fundamental changes that seemed to be occurring. That low activity in the frontal lobe was corrected with these various treatments, but there was this interesting area that no one really knew very much about, the subgenual cingulate, and its anatomical designation is area 25. That's Brodmann's designation of the term, certainly not mine. Kind of like, was it area 41? And this is Brodmann's area. But that this area actually turned down with successful treatment. And in fact, we had noticed that if you took Prozac or paroxetine and you didn't get better, you didn't get the change in terms of decreasing, turning down the activity in area 25. We saw this, and you can just follow this along with placebo. And we started to look at the literature that even things like transcranial magnetic stimulation, magnetic stimulation applied actually to the frontal lobe, also could turn down area 25 by getting clearly into the circuit. Drugs were turning down 25 and turning up frontal lobe, which you can't see here. TMS was aiming at the frontal lobe, turning down area 25. And even something as non-specific electroconvulsive therapy, which in essence is like turning off the main switch on your computer, and when you turn it back on, it kind of works again, a really non-specific kind of rebooting, that if you looked at the brains with scanning of patients before they had ECT and after they had ECT, it was reported that, well, boy, this is remarkable. Their brains have low activity, and the better they do after ECT, gee, the worse their brain is. Everything goes down. Well, you know, the glass half full view of that same data is that, well, you got a lot of extra stuff that maybe you didn't want, but just like with TMS, placebo, SSRI, other meds, that actually area 25 was turning down as well. So somehow, not the idea of just an absolute change in a bunch of regions, but somehow the coordinated interaction of these regions maybe was most important. And at least to my eye, since it was my party at this point, area 25 seemed to be a fundamental change. Now, it was interesting, and this was part of the project we did with the Dana Foundation, was we were interested in the component pieces. You know, neurologists are about which function lives where. So where's the sad part? Where's the sadness? Mood disorder is fundamentally a disorder of mood with a lot of other stuff. That's the defining term. That's why it's called a mood disorder. But if you actually take even healthy people, you have them recollect a personally sad memory and actually scan it, look at how brain blood flow is changing going from neutral to sad, area of the brain that has the biggest change in activity of every voxel in the brain was area 25. And at the same time, your frontal lobe turned off. So this idea of, we'll call it a pathway, although there were a number of regions, the relationship between 25, a limbic structure, and frontal cortex, a cognitive structure, were vitally linked. And everybody who's had a sad experience or listens to something, you know, sad on the television, you turn inward, you become very inattentive to everything else going on around you until the story changes, your attention is diverted, and the mood dissipates. We're wired to have emotion and thinking interact at all times. Well, it turned out that this area 25, even though there's no naturally occurring lesion in this area, you don't tend to get tumors there. If you get a vascular event, it doesn't affect this area uniquely. Everything we know about it actually comes by inference from wiring diagrams and studies in animals. It's a visceral motor center. It's part of the brain that when you stimulate it in animals will evoke sad cry in monkeys, like when you separate a monkey from its mother. But it also sets off autonomic responses, talks to the hypothalamus, it regulates circadian rhythm, sleep, all the things that are also at the core of major depression. And it turns out when people did start to look at the microscopic anatomy in the brain of depressed suicides, they found that there were actually something funny about the glia, not everywhere, but also right here. So there's this convergence of treatments need to turn this down, sadness is about turning it up, something's wrong with the wiring nearby in the glia in this area. And so if one says, I've got a very elaborate network of regions I want to do something to, but boy, this place seems to be a little more fundamentally important, is there a place in the brain that I could modulate using the technology that had developed with DBS to kind of change the state of that circuit? Where could I go? And in fact, if you just look at this very high resolution MRI that allows you to look actually at the white matter right near area 25, and here you can see in treatment-resistant depressed patients, this area is overactive. You can actually see there's almost like an intersection of tracks right under area 25 that actually will connect to the emotional parts of the basal ganglia, to the limbic structures and hypothalamus that regulate circadian rhythms, and connect anteriorly and dorsally up to frontal cortex. It's a very, very funny place where there's a convergence of all of these systems. And we said, and I spoke with Andres Lozano and said, well, can you put an electrode there? And surgeons always say yes. It's good about surgeons. You always want surgeons. They'll always say yes. And we did exactly what Mahlon described, using exactly the pioneering work done with the same kind of technology of deep brain stimulation. And the goal was to turn down area 25. And if we turn down area 25, it would have impact through the circuit on all of its co-conspirators. And in fact, we can target it easily. It's much easier than what they do from Parkinson's. You can see it on an MRI. They have the same kind of frame. You can see where we want to go. We place an electrode on each side, hook people up to the pulse generator, and we did this in a group of patients who basically had failed everything. And that was done, first patient in May of 2003 in Toronto. And I'll share with you what happened, because again, the dogma in depression is whatever you do is going to take some time to get better. So we know where we want to go. We know we want to turn it down. The Parkinsologists told us that a certain frequency would turn down this system. We'll do exactly what they did. We'll just put it in our place and not their place. And we'll make sure that because of these connections, we don't want anybody to have a complication, like maybe people's heart rate's going to go crazy there, or autonomic function is going to be affected. So we kept patients awake, just as they do with Parkinson's, although we had actually no idea what would happen when we turned it on. And I'll tell you, we were surprised would probably be understatement, but I'm trying to be understated. And the first patient on the first contact, actually the second contact on the first side, the patients basically, we're starting now, anything changes, let us know. Starting from 0 voltage, moving up slowly, suddenly said, did you do something? I have this sudden, intense feeling of immense calm. And as she went to try to describe what was happening, it was a relief, it was a quiet, it was extremely hard to describe. This is a very non-cortical phenomena. It's like a change in internal state. It's like, how do you have that gut feeling? What is that state in the pit of your stomach? To have it suddenly go away, what would that be like? Well, different patients, as we did subsequent patients, described it in different ways. It was unique for every person. It's not like watching the tremor stop. It's subtle like a brick, but it really is selective for each person. And to summarize, neurologists are splitters, not lumpers. The psychiatrists get to have all the nice narrative. But in fact, it's a dissipation of the visceral part of the illness. Whether it's the dread, the void, the mental heaviness, patients, literally describing that grayness, that weight kind of lifting. And it would be dose-specific and contact-specific. And you could turn it on, and if you got it, if you turned off the contact, they would kind of go, oh, maybe that wasn't anything. But there was an abrupt cessation of this core part of the illness. And in fact, it was like a change in interoceptive awareness. A change in the body state, not in thinking. In fact, to describe it was really hard. What was wild is I would say 10 to 15 seconds later, while they're trying to describe this and we're trying to listen, patients became more spontaneous. These are patients that are usually quiet, very uninitiated, a lot of apathy, very slow, very frightened, and suddenly became sort of engaging, more motivated. They spoke faster, as though their psychomotor slowness changed. You could test them by their motor speed and watch that change. But they described things as though they were just more interested, had more energy. They're in an operating room, can't really do anything, but describing that if I was home right now, I might actually be able to clean my garage. I haven't washed the dishes in a week. Just basic activities. A sense of initiation that many depressed patients who respond to other treatments all describe. The really great news was that nothing bad happened. So we did not have to abort the experiment. We did not have to stop because of changes in autonomic functioning. And so we could identify which electrode seemed to be best with the plan that that would be what we'd stimulate chronically.
Just to give you an example of what patient said, he described his depression as this gnawing agony, a painful self-loathing that consumes all of his energy. His patients describe a state, I am so turned inward that I really cannot attend to anything else. And when he had an acute change, he said it was as though I just suddenly shifted from a state of all-consuming internal focus, just realizing that there were just a number of things around to do. And he then went on to make this remarkable statement. Sorry, I think it's a remarkable statement, and below the, hold on. It's as though I'd been locked in a room with 10 screaming children, and parenthetically, he had 5 children. And this was like his kids bringing this noisy, unstoppable, this was like what he lived with constantly. No rest, no escape. He goes, whatever just happened, the kids just left the building. So again, not something a neurologist is used to hearing. Not sure how I localize that totally, but speaks volumes. As an imager, the bottom line was, did we do what we thought we were doing? And the good news for me was that we could actually inhibit area 25 just as we'd hoped. And we also saw changes in the hypothalamus. We saw changes in the ventral striatum, these areas in the emotional striatum. And we got correction of the frontal lobe. So we actually hit this whole network by chronic stimulation in this location. And as you can see, these patients did extremely well. We published the first results a year and a half ago on the first 6 patients. These people didn't get better. They're well. These are people who are disabled, that these 4 people have gone back to work. The numbers on disabled people after a year of disability is really bad. And these are people that are picking up not necessarily what they used to do, but certainly taking on, in some situations, going back to their previous jobs, and others, new activities. We've now operated on 20 patients in Toronto. We have data to speak about on 15. And we're seeing, more than half meeting these response criteria at 6 months, and actually a better response out to a year. And it doesn't look as though it's accumulating an improved response over time. It's just you catch people, they have a bad day, they come in, they get their Hamilton score, and it bounces around a little bit. It looks as though if it works, it works. If it doesn't work, it doesn't. And what we don't know is who the right patients are, which is extremely important. I'm just going to finish with what a patient described, because people have often asked, well, is this a mood elevator? Again, a history of stimulation in terms of affect has been intracranial self-stim. Stimulate, get a rush of some unknown type, and that rats will bar press for that stimulation. This is not that. Patients are very adamant, as this patient described, look, you didn't give me anything. And that was actually his tone. He goes, but you took something very profound away. And to him, it was this constant void, like he was in this vortex being sucked down into some deep pit. And it just vanished in the operating room. And he goes, now it's like instead of being in the Grand Canyon, I'm up on this ledge. And I'm actually part of my own recovery. I'm not better yet, but I can actually participate. We're optimistic. We're optimistic enough that we now have permission to do this at Emory. We have permission to do another 20 patients. We're going to exploit every possible means to try to understand what we did mechanistically, and importantly, to do it in a controlled way, to have blinded periods, to actually have periods even of discontinuation after 6 months to see if it holds, but also to avoid the thought that this is some kind of very sustained placebo effect. But we're encouraged that this location and this guess wasn't a bad first guess. We certainly need to understand the nuances of this, just like they did in Parkinson's. And I think the real critical step now is, who are the right patients? How can you meet all these criteria and still have people that it really doesn't work on? We have a problem in terms of what our definition of treatment-resistant depression is. But I think it's giving us some important new insights into depression pathophysiology. But I think the money will be on if we and others can do like experiments. There may be, like in Parkinson's, other places in the circuit that are equal, better. I think, a side effect profiles. Maybe one area actually is faster or more sustained, or you could stimulate there and turn it off. I mean, there are all kinds of questions we can ask experimentally, but that's where we are. And I think we wouldn't be here if we didn't have the guidance and the work of people like Dr. Delong, who basically blazed a trail, got rid of all the low branches and kind of created an environment for us to test these principles. And I'm grateful for that.
We're a little bit running behind, so Brian, I'm going to put you up there. We have time for, while you're getting your thing hooked up, one or two questions if there are any. Yes?
Let me ask a fairly general question. We've heard from all three of you that all these problems are in circuits, yet from both of the last two speakers, we've heard of interventions at particular places. Now, if you're dealing with a circuit, you would think that you could intervene at other places in the circuit. Is that something that you are working on or thought about?
Absolutely, and other people are thinking about this as well. So in depression, people have targeted the nucleus accumbens directly. And theoretically, 25 talks to nucleus accumbens, and I think the data will have to evolve to know, are all components of the syndrome alleviated the same, or does modulating at one target impact the next steps along the circuit in equal ways? So you may be able to get into the circuit, but side effects may be different, which was certainly the experience, I think, or the growing experience in Parkinson's.
With Parkinson's, and I didn't make that as clear as I should have, that the STN is a good target, but the pallidum is equally good. In fact, we're doing a randomized study that so far they haven't stopped us from doing because we think it's an equivalent target, and that's what we set out to show. So I think your point is exactly on target, that it looks like we can modulate this over across many nodes of the circuit. And that's the question, is which one might be optimal for certain patients or free of certain side effects or complications. So that's where the work yet needs to be done.
Yes, ma'am.
In the same way that you talked about variability, patient responses, and being able to relate it to changes in activity in area 25, did you see something of that nature with the stimulation?
In terms of the downregulation or not?
Yeah, in terms of relating how much change in activity there was to the efficacy.
We don't have an absolute to be able to say. It may be that non-responders actually didn't meet a threshold of percent decrease in 25. I mean, even non-responders had decreases there, but didn't necessarily have decreases remotely in the circuit. So the question is the break in the circuit in a different place, and is a particular target need to take advantage of actually what the wiring is in a given patient? And that's, Dr. Mobley talked about the diffusion tensor work. And instead of actually looking for structural gray matter abnormalities, maybe we ought to be looking for kinks in the wires. And
okay,
he's ready.
Great. So also, it's really a pleasure to come and speak with you this evening. And you're in luck. First, I'm the most junior person up here. So first, not only did I have to carry all their briefcases in. No, that's not true. But I'm also the only one who probably made 10 slides. Okay, so listen to the directions. I'm just kidding.
I made 12.
A few things. Part of what I do is to try to generate new ideas and develop technologies that we commercialize and put into people. So it's very important that I disclose that I've consulted or worked with these different companies to do that. We've licensed technology through the University of Pennsylvania to some of these companies, so there's a potential bias. I'll leave that to you to judge. I try to be my own harshest critic. So now where we're going to go is into the cortex. So you've heard about networks in movement disorders, where there was a good suspicion that that's where the money was for these disorders, and Dr. Delong pioneered these techniques to really study these circuits and figure out how they worked, with tremendous inspiring results, and it's also a reason why I'm in this field. Then we've heard from Dr. Mayberg about circuits where it wasn't really clear where they were, but there were techniques to find this location, at least one spot in the circuit to go for, with also amazing results, and it's a story in progress. Now I'm going to take you into epilepsy, which is a disorder primarily of cortex. And the problem we have is that we can create epilepsy anywhere in the cortex. So it's a matter of not where the exact circuit is, but where the circuit is in this particular patient and how we deal with it. I put this slide up just also to acknowledge that you can never do this kind of work alone. I work with a large team of people, collaborators in various departments at Penn, at Georgia Tech, at the Mayo Clinic. I have their names up here. They'll be posted on the web. And without them, I'm just their ambassador this evening. So epilepsy is an enormous problem. One problem I have is that I'm speaking after Helen because depression, of the disorders that are more common, that's one of the only ones that is. So she's taken a little bit of my thunder. But 1% of the population, about 60 million people worldwide. A third of people with epilepsy, and epilepsy is defined as abnormal synchronous electrical activity in the brain that changes behavior. If you have a seizure coming from your visual cortex, you'll see bright flashes of light, phosphenes. If it's from your motor area, your hand may contract uncontrollably. If it's in the temporal lobe, you may stop, stare, chew, pick at things. If you do that in the wrong place, it could be awful. I had a patient do that in front of a police officer who stopped him. He reached out unconscious and did this to the policeman's vest, who promptly smacked him to the ground, cuffed him, and put him in jail for assault. It can be a tremendous burden to people. People with epilepsy die more often. They have more disability. And the majority of people with epilepsy are not people that are severely disabled if you were to look at them on the street, or like Dr. Delong's dystonia patient. They're people like us who may not have any outward signs of their disorder. Just out of curiosity, how many of you here have seen somebody have a seizure or know somebody with epilepsy? That's incredible. So the vast majority of people in this room, and that's a testament to how common it is. A third of the people with epilepsy are refractory to medications. We can operate and palliate, sometimes cure, maybe 7 or 8%, but that leaves fully 25% of people who can't be controlled on any available therapy. It's an enormous problem, and one that really demands our attention. People who are controlled on medicine, they have side effects. They would rather have other alternatives. Not everyone, but the vast majority of people with epilepsy have side effects. There's a desperate need for new therapies, and that's why I got into this area. I was in practice, actually in the Hopkins system, and had an epilepsy outpatient practice concentrated with refractory patients, and I had one of two decisions: either to find a hobby and try to forget about the problem during my time off, or to go back into full-time academics to work on it. And so with this kind of inspiration from these folks here, that's why I'm in the field. What are their options? We can try experimental medications, but we know if you have trials of 2 appropriate medications pushed to high doses for your epilepsy syndrome, it's a heterogeneous disorder. There are many potential causes. 2 medications pushed to high doses, documented blood levels, you fail those, you fail a combination of 2 together, your chances of being seizure-free on medicine, 10% or less. Okay? You might gain a few, but it's a pretty terrible thing. So the field of epilepsy surgery really grew up out of the background that Dr. McKhann gave you, with noticing that lesions in particular parts of the brain could cause seizures. Electrical stimulation in the operating room could cause seizures. And sometimes removing lesions, like abnormal blood vessels, cavernous angiomas, things like that, could stop them. Again, the idea, although when I was training, there was this idea that epilepsy came from a spot. You had to find the bad spot. You had to map the brain around it to make sure you weren't going to remove something important like speech. And if you took out the bad spot, their epilepsy would go away. But it doesn't. It turns out that if you take out that spot, unless you take an enormous piece of tissue, and I'll show you an example of such a surgery, the best surgery for some people, the best treatment for some people, unless you take a huge piece of tissue, it'll go away for a while, but it'll come back someplace else, someplace connected. The whole idea of networks, and now it's well accepted, and there are people in this room like Dr. Gale who've done enormous work in this area that have demonstrated that networks are really important in epilepsy. What are the common causes? The most common cause is We don't know. We call it cryptogenic. That's a great medical word. We don't know. It turns out, though, when you resect tissue from people with cryptogenic epilepsy, you very often find cortical dysplasia. You find neurons that aren't formed or connected right. Kind of a hint of a circuit disorder. Of the most common known causes, focal lesions, injury, the most common, head trauma in this country, automobile accidents, etc. And there are a lot of genetic causes. So the most common MRI scans in our patients look like this: normal. You might have somebody who had a bad encephalitis or a prolonged febrile seizure in childhood who has this normal hippocampus on this side and a shrunken one and with bright signal on this side, mesial temporal sclerosis. So great idea that was discovered. Sclerosis means hard. It was hard in the operating room. You remove that focally and they get better, right? No. Take a look at how nice and plump this temporal lobe is over here. Look how shrunken that whole temporal lobe is there. It's not just there. It's a network. And if you do fine functional and structural imaging, you can notice that the thalamus on that side, you can't see it here, is also abnormal. That the limbic system, the cingulate gyrus, and other connected regions are abnormal. So just removing that doesn't help. Large vascular lesions also can be causes of epilepsy. So what do? I apologize for showing these before dinner, but epilepsy and its surgery and evaluation provides a window and access to understanding cortical function like no other disorder. In major centers where epilepsy surgery is done, these patients are not only there for treatment, but they're also pioneers. Because at virtually every major center where there's good surgery going on, there's good research going on. And these patients allow us to record from their brains, from electrodes placed in the brain, to stimulate, record, try to understand and interrogate the networks that are involved in generating their seizures. What are our tools? These are depth electrodes. They look like shish kebabs a little bit. They have 6 or 8 contacts on the end. And when they're placed in this Montreal technique here, they fan out like this. Their other centers have techniques named after them. This is artifact here too. We don't put number 2 pencils in people's brains. These are quite small. We have grids and strips of electrodes that are placed over cortex like this. And then for people with mesial temporal lobe epilepsy, like I just showed you, that scar, we take a brain that looks like this, and we take it in, turn it into a brain that looks like that. We do a temporal lobectomy, and we take out the middle part of the hippocampus as well, the anterior 2/3. That's state-of-the-art surgery, and for these people, the very best therapy available. If I had that, I would have this operation. But there probably is a better way. We know that these circuits don't usually occur right away, but they occur over time. A typical story for epilepsy of known cause: somebody has a head injury, usually associated with loss of consciousness or amnesia, and then a latent period. A period of time when they've recovered and they're fine. It may be months, it may be years, it's often 10 years or more. And then they develop seizures. And when you take a look carefully at the pathology of their brains, you notice that they're missing certain cell types. There's decreased metabolism in these regions, and there's a network of abnormal tissue and connectivity. So what do? Well, if it's in the neocortex, not in the temporal lobe, or neocortex outside of the temporal lobe, we might do mapping. So here's a patient in yellow who had a cortical dysplastic lesion, a tiny little like whiff of smoke on the MRI scan that was abnormal that a good radiologist picked up. The patient was implanted. We recorded the seizure's onset in red. It spread very quickly to the purple, then to the green in full-blown seizures. The patient had these little high-frequency oscillations in the bright green. And this is just motor speech. We do more complicated mapping than this. But this is very crude looking compared to what you saw from some of the previous studies from Dr. Delong's diagram. So we began to ask questions like, why is it that we have electrodes that are spaced like this and look like this? Why is it that typical EEG for intracranial EEG is recorded from 0.1 to 70 or 100 hertz, when basic scientists in the laboratory have been recording out to kilohertz? How fast do individual cells fire? Really fast. And we began to get answers to these questions, like the reason the bandwidth on the EEG machine, traditionally and in commercial equipment available today, although it's changing rapidly, is 0.1 to 70 or 100 hertz is because that was the limit of the frequency response of pens on paper for paper EEG machines. Why is it that these contacts are shaped like this and this size? Because that was deemed to be safe and reasonable 50 years ago. So we've started to ask questions at our research group and a number of other research groups to try to explore how do you figure out these networks, because you know something? This probably isn't all that's going on in this patient. Well, in our group, we have kind of an interesting mix, and it fits this multidisciplinary issue and theme that actually Dr. Delong hasn't spoken as much about, but if you, I've worked in the same centers and seen that he doesn't just have neurologists who see patients in the clinic, but he has and is himself someone who interprets neural signals down to unit recordings, that analyzes them statistically, that uses mathematical and engineering techniques to understand how they all fit together and how they're synchronized. And that's what we do in our group. We're a mix of engineers and physicians, and I have postdocs who are both PhD engineers and physicians. And we measure signals that come out of these electrode contacts, and we've started to use other contacts similar to what's been going on in the Parkinson's work, where we look with microelectrodes and record multi-unit activity, clusters of cells. And we're trying to answer questions like, how can you tell, what are the earliest signs that an area of the brain is involved in a seizure? How does it start? How does it spread? And can you measure the connectivity from one place to another? So in this study, for example, this is 8 hours before a seizure, we chronicled that there were a group of physiologic changes, and this is not prospective or randomized, but just an observation, that there were bursts of energy that measured that corresponded with this abnormal epileptiform activity hours before seizures. Beginning about 2 hours before, there were these little mini seizures that may last a couple to 10 seconds. We call them beta buzzes in the clinical EEG, but they're chirps by engineering standards. That means they frequency modulate, like when a train goes by, they go boo, like that, or boo, or up and down. And then beginning a number of minutes, usually about 20, but sometimes up to an hour before seizures, you see a ramp up of energy before the seizures in blue that you didn't see far away from seizures in red. These, in retrospect, are relatively crude observations. But now that we're looking more carefully at much broader bandwidths, up to 32 kilohertz, down to DC levels, now that we're looking with microelectrodes in data that we haven't published yet, actually, the papers are in progress, where we actually are measuring multiple units and aggregates of them, we're seeing very different things. And I'll talk about that in just a minute. But with this quantitative approach, there's already been a big change. Now, there are some different paradigms here. Depression can be an episodic disorder. Movement disorders, Parkinson's, can fluctuate, but it's usually a pretty fixed disorder. But epilepsy is very episodic. And we've actually taken a lot of our lead from the cardiologists. So if you have something like a cardiac dysrhythmia that comes once in a while, what you want to do is have a device listen, and when it sees the sign of something going wrong, interpret it and react to it. That's one approach. There is another approach with the same devices that are being used for depression and for movement disorder, same company, where they're looking at areas in circuits that have been defined in animal models and humans, like the anterior thalamic nucleus. And this is a stimulator, I think same electrodes you're using, aren't they, Helen? Probably. And this is a trial that's going on, multi-center, 200 patients this year being implanted with the same device to do chronic stimulation in the anterior thalamic nucleus. And the trial, I don't know any results yet. We know from pilots that it's likely safe. We don't know that it's effective, but they've passed their midway point and the trial is going to continue. So that means they must have enough evidence to continue the trial. This device here is a responsive device, like a cardiac defibrillator. Here's a recording from an actual patient tested at the bedside. You can see the seizure begin, this rhythmic activity that spreads, the device senses it, boom, it stimulates, and the seizure's gone. Does it mean that it's safe? doesn't mean that it's safe. Although there's a trial of 50 patients so far where there have been no complications. It looks like it's probably safe. And the FDA has given approval to do a pivotal trial, and there are 200 patients getting this device now. There are a lot of questions. What does it mean to stimulate these seizures? Early data shows that, remember, there's plasticity in these networks, right? So, for example, whenever you store a memory, your cells change in the way they're put together, right? You have to physically store that memory someplace. When you stimulate, there may be changes too, and what they're seeing is that the seizures are changing a little bit and migrating a little bit. But the initial safety trial, not designed for efficacy, of the responsive stimulation device showed 50% of the patients, a little bit less, about 45%, had a 50% or greater improvement in the seizures. Does it mean it's effective? No, it's not an adequate clinical trial. Does it mean there's some proof of principle, enough maybe to move forward? Yes, but it's first generation. Defibrillators are on their 7th generation right now. But so I started to say we're finding out more about how these seizures are generated and about their networks. And what we're finding is we have a problem with scale. And this is something that we heard in the first talk. We're looking at brains with 10 to the 11th neurons, and we're putting grids over a portion of that, enormous networks. But look where the activity is. Some researchers have shown that in models of epilepsy, and in humans too, you can have mossy fiber sprouting in temporal lobe epilepsy. You can have loss of GABAergic interneurons. You can have other cell changes that are only seen on this level, but we're not recording at that level. We have these big fat discs that are recording at a much lower frequency. Is cortical columns the right spacing distance that we should be looking at? Maybe, we don't know. How about individual cells? So right now there are several groups around the country, and the majority of the work in our collaborative group has been done by Greg Worrell and Matt Stead at the Mayo Clinic, where we have microelectrodes in place, and we're seeing some very exciting things. Long before there are any seizures that are detectable in the intracranial electrodes, they're seeing micro-seizures. They're seeing abnormal firing in aggregates of units, little pockets of cells that seem to be firing synchronously. And the question is, do they come and blend together and get synchronized? Could we stop the seizures if we knew really far in advance? These questions are open, and we'll have to read Greg and Matt's paper to find out. So stay tuned, okay? The other issue is the frequencies, and I mentioned we're going out to 32 kilohertz rather than just down in this small area. Well, what's new? How are we going to map these networks? What kinds of technologies are we going to use? And we have some ideas in our lab. The first is, it would be great if there was a way to be able to interrogate regions of brain without opening big holes in people's heads and putting the electrodes in, because there is nothing more horrible than making somebody go through a surgery like this, seeing the seizure begin on the video when we monitor them, and about 30 seconds later see it march in from the edge of the grid. It means you missed. Right? So we ought to have a way to do that. Well, as part of the work I'm doing, my Dana grant is actually on brain-cardiac interfaces. And so why not imitate our cardiac colleagues a little bit more? If you want to know what's wrong electrically in the heart, you take a catheter, you stick it up into the heart, and you stimulate around the bundles and try to precipitate dysrhythmias. So we're taking catheters and we're putting them up in blood vessels in the brain. We're not doing this in humans right now. We're doing it in pigs, actually. And we're stimulating, trying to interrogate networks. Can we create echoes that you can see at other places? We do craniotomies also. We put these catheters in. We stimulate and record. We cause a seizure on the cortex, recorded here and recorded here. And can we develop some kind of brain electrophysiologic testing? So maybe. The next thing we do is we have computer-controlled stimulation to map these networks for function. So patients play computer games while they're hooked up to these intracranial microelectrodes, and they do cognitive tasks, and I'll get to the punchline. Like this virtual reality game, which tests different things. And patients, while they're doing this, we stimulate different regions and try to knock out certain areas and see where the functions are. So Dr. McKhann asked me in closing to say where I thought the future of all this might go. So this is a little far-fetched, but suppose you have a network disorder, suppose it's epilepsy, And we don't really know where it is. Well, you get some nice diffusion tensor imaging like we saw from Dr. Mobley early on, and it kind of maps pathways, and we look for incongruities with computer pattern recognition areas. We might find some abnormalities. We can test them with functional imaging, and then perhaps you get an implant which interrogates these networks and maps them. Then you go up to Stanford or one of the other universities that's doing neuromorphic engineering, where they can make chips now that can emulate neuronal encoding. There's one that I've seen that actually was published in Scientific American by Kwabena Bohan, who had the office next to mine at Penn, where there was a video image captured on the camera. It went to a chip which was built like a retina and put out the neural code that your brain would recognize to see that. So perhaps you get fitted for a neural prosthesis, and then you have a brain-computer interface where cells are grown on the interface between the silicon, and you have your repair done. It's a little far-fetched, but I can dream. I wanted to acknowledge the sources of my support, and thanks for your attention. Sorry to talk so much.
Yeah, so over here, you were...
Yeah, I wanted to relate the experience and ask a question. A few summers ago, our family was on vacation, and we were in one of the sort of the reproductions of a frontier town. We were walking through, and there was a doctor's office. And in the doctor's office, there was a medical journal. Now, this was a genuine medical journal of maybe before 1860, and I was sort of paging through it, and melancholia caught my eye. And I started reading about it a little bit, and melancholia sure sounded like depression to me. And the script went on to say that we don't know what's causing this, but give the patient just about anything and tell them it's a medicine, and in general they turn out okay. So,
and your question is?
Yes. With respect to depression again, I've had bouts of depression personally, so I know about it a little bit. It's based on, I think I've read a lot that the...
coming to the question.
Yes. The synapses have a problem in firing. I've taken SSRIs and so on. And I'm just wondering, rather than causing a mood problem, I'm just wondering, the synapse is not firing properly. Why doesn't that just make me stupid instead of giving me mood problems?
Well, I think it's the same reason that when you damage your motor cortex, your nose doesn't itch. That it's about location, 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 but location isn't just about one spot, one behavior. If it was, your brain would be as big as a refrigerator because you'd need an awful lot of places for all the functions to live. And that's what's so interesting about thinking about circuits. And I think with Brian's comments, not just linear relationships. I mean, I think the basal ganglia has some linear relationships, so that one area talking kind of, point to point has been one approach, very successful, whereas what Brian's talking about is there's a much more almost chaos theory requiring way to think about how the systems behave. So I think that different synapses, serotonin hits many, many areas of the brain. And it may be that the only ones that you need to modify the behavior are really a very small percentage. But there's no way to deliver the drug just where you want it. So the idea of a circuit is not just about electricity, but is about anatomy, the connections, and the chemistry that regulates that particular circuit. All things are important. And so I think that, a circuit is not a circuit, it's not a circuit, it's the circuit where.
Let me make a comment, and I'll be interested to hear my colleagues as to whether they agree. My comment is related to the challenge. The idea that the brain is organized as circuits, you've heard a lot of evidence for this, and I think we could take other areas and give you that evidence too. The problem is, how do we get at them? How do we figure these circuits out? Now, Mahlon was able to go to the monkey and look at particular areas and put electrodes in, but as Brian was saying, that's actually rather crude, and you have to know where to look. And these other circuits, Helen doesn't know where those circuits really are. So how are we going to get at this? How are we going to crack this problem in the human? Do we have to go to the primate first and then say this is where we know how to look? Or are we going to use your techniques and say, okay, we'll get to a lower and lower discrimination, and when we can get at the human brain, we'll do it? What do you folks think? Mahlon, you started all this stuff.
Well, I think this is a big challenge, and I think even the work that we do in subhuman primates doesn't necessarily map perfectly onto the human. And in some ways, I think the human is the best model for understanding the human, as curious as that sounds. I think the imaging techniques are getting certainly to the point now where we can start to reconstruct or examine some of these connections, tensor imaging, and sort of see where these points are connecting. And some of the modeling that David Eidelberg has done with PET gives us another approach for looking at systems and correlations, areas that are correlated or related. I think there's no one technique that's going to do this, but the combination of approaches. And now we also have greater access to the brain in the course of these procedures that we carry out for sort of routine things like Parkinson's and other disorders. There's the opportunity to gather information that has heretofore not really been taken advantage of, but with the newer techniques we're able to use almost every operation as an experiment and gain information that would have been impossible before.
Guy, I'd like to ask, is there any controversy involved here? What do psychologists say about deep brain stimulation? Are we presenting too optimistic a point of view?
I'll take that. I mean, I think that, again, what I've noticed as a neurologist using technology that I'm not an expert in, like imaging, and working in a field in which I'm not an expert, namely psychiatry, I've lived with the mafia of camps. And I think, again, the take-home message is no one disciplined knows what they're doing adequately to actually stake claim. And this kind of notion, and people approach a problem from the degree of their own expertise. And I've noticed with the brain stimulation experiment, again, I want to emphasize what Brian said, this is not ready for prime time. This was a proof of principle. Is this circuit relevant? Can it be modulated? And is it safe? And now we will go forward. And it is definitely not a treatment for all depressed patients. If you can get better with therapy, you should have it. If you can get better with drug A, drug B, you should have it. The issue is that we don't know which person needs what treatment because we don't fundamentally know if depression is depression is depression. And the word is a bad word, needs to be changed. And it may be that people who have a malignant form of the disease need to have another name for it. And I'm hoping that Mr. Sapphire will provide one since that's his great talent. But at the same time, I think that delineating the system is critical, but I have noticed that psychologists, people have been very flip when they've been asked about the results of our study. If you talk to a psychologist whose only avenue to assist patients is to talk to them, they assume that we haven't tried that. That is a gross presumption and actually fairly insulting. And so I think people reveal their own biases in a way that I think we always have to monitor. I think we do bring biases to the table, and we have to be self-monitoring that to ensure that we are experimenting on people who have exhausted all options and not assuming that one size fits all. So I think that, you know, again, I found that people who, I noticed on a blog site right after our paper came off, that someone said, who the hell does this Mayberg think she is, putting electrodes in people's brains? Is she, you know, what does she think she's doing? And then there were 5 responses at that day to the blog. Well, so what are you saying is wrong with people who've been disabled, who now actually have a life? What's wrong with that? So I think, again, you can't project your own experience or your own perspective alone into the mind or the disease of someone else. And I think that we'll just need to do our job and see.
I would just say in answer to both of your questions that concrete things that we could do to get at this problem and at the same time deal with controversies are to make sure that each institution that's doing these kinds of procedures and this kind of research, one, be required to communicate with others, that there should be some certain standards of practice. Certainly every institution has an IRB that regulates these kinds of things, but there should be shared data, and there should really be an effort for each center to be working on the problem together. So in my field, centers have different equipment, they have different data formats, they have different protocols. They're all based upon tradition, but not on rational thought. And so, I mean, it's rational thought, but you know, it was the way Grandma did it when she taught me. And so, I think that, oh, sorry. You should see him in that sundress. No, sorry. But it's true, we have to get some organization, some standardization as well.
I kind of endorse that I would not want anyone to take away that we don't have involvement of psychopharmacologists, and actually what will ensure the success of what we're doing is once the brain comes back online, it has to be retrained. It's in essence what happens if you've had a bad hip for a year and you get a hip replacement. The surgeon doesn't just say, oh, wow, successful hip replacement. If you don't have physical therapy, you will not be better. So once the brain and the rhythm is changed, then the brain needs to be retrained. And a surgeon doesn't do that. A skilled therapist does that. And our patients that have the combination, they seek out therapy in a way that was not accessible when their brain was in this malignant state.
Dean Newton, you want to wrap up?
Yes, I'd just like to say on behalf of Bill Sapphire and the Dana Foundation and the College of Arts and Sciences at Syracuse, I hope we can agree that the presentations and discussion here present as eloquent testimony as would be possible to the critical human importance of kind of spanning the moats among the academic disciplines in order to create new sciences and new technologies. I'd like to ask for you to join me in thanking this eloquent panel.
Okay, team.
Thank you for joining us, and good night.